apresentar as perspectivas, que são perspectivas biotecnológicas no controle do greening, né?
We'll talk about biotechnological strategies for controlling greening. The strategy of biotechnology at Fundecitrus has three areas. One is focused on HLB greening with three topics that have been working with along 3 years. A plant that is repellent to psyllid. That's where I'm going to be more focused for most of the time in my presentation. Also, we have the lethal myrtle, picking up where the previous speaker left off, and we're going to be talking about genes for controlling the bacteria. So when we use this biotechnological strategy, we have also the reduction of the limonene in the fruit for controlling the fruit fly, and we have found tolerance characteristics in the lab, but we're not going to be talking about this. And also we have beneficial qualities of the juice. That is also another aspect of biotechnology. Fundecitrus decided to invest on genetic engineering or biotechnology in 2008 due to this challenge of controlling and managing greening. We know that there is no genetic resistance against greening or Liberibacter or HLB, so there's no possibility of genetic breeding conventionally done for controlling this disease. We have seen, yesterday and today, people talk about no use of rootstocks to control it, as source of resistance. So biotechnology will bring characteristics from out of those cultivars to be incorporated to the citrus to develop resistance. Can be resistance, repellence, as has been said here before, and to the citrus grower, we need to have a low cost of for management and the absence of genetic because of this lack of genetic resistance. So we started this program in 2009, and then we incorporated technology of mature tissue for as a relevant characteristics of this product, because when we have a plant, this plant will go to the field and will produce in 2 years. So this technology has been brought from Spain, and then it was perfected by the IAPAR people in Paraná. In 2012, first plants were developed, and then there was a challenge in 2013 that was to have a methodology proposed to the National Biosafety Committee, and it included testing plants in the field. And then we kept flowers and fruit of those plants in the field, plants that were genetically modified. So norms were set forth, and today we have a protocol set forth, and that expedites us in our field trials. And some plants are here in January 2016, and we follow up on their development. Here, the main project, that is the repellence of psyllid in guava trees. The work carried out in Vietnam in the beginning of the century showed that monocropping for citrus tangerine presented high levels of greening right after planting and high presence of psyllid. For citrus growers in Vietnam saw that citrus together with guava, what didn't happen with banana or mangoes, in the first years of planting there was a very low incidence of greening and almost no psyllid. This field observation then was studied in Japan and Australia in a research project, and then we could see that in the first year we had the incidence of greening and psyllids in those growths in a much smaller incidence. And then the second year was capped, and as of the third and fourth year it was lost, probably because of this difference in size of plants. Then there was a mission by Fundecitrus, captained by Professor Beauvais, visiting Vietnam taking people from the industry and research for them to understand what happened in the field. And they were convinced that there was a strategy that could be addressed with the biotechnology, not using guava in the field. And so the first remark was that maybe there was some component of the guava that interfered in the citrus plant in the field. So we studied the attractiveness or repellence of the guava plant, and we concluded that the guava was repellent to psyllid. So Dr. Walter already talked about the olfactometer, and we had this experiment carried out, and insects then can have this odor field from plants and odor field from the pure air. So if they choose the plant is attractive. If they choose the pure air, this plant is considered to be repellent. So the guava was repellent. But the objective was not to use guava planted together with citrus, but rather use this repellence as a component. So we determined the component that was present in a guava plant that was repellent. We have some literature data also to help and some studies carried out at Exalc and then Fundecitrus. And we compared citrus, that is a monoterpenic plant with limonene as its first terpenic compound, while guava are sesquiterpenic. They have terpenic compounds, but in a different category. And out of the main sesquiterpene in guava plants, we have the caryophyllene, and that is what prevails. You know, there are volatile compounds released by guava plant that was studied as a repellent. So we carried out tests with the olfactometer with this pure compound, and a certain concentration it showed to be repellent. So a pure compound present in a volatile component of guava plant showed this behavior in face of psyllids. So we then had to prove that a plant producing compounds like this in a high quality would also be repellent to citrus psyllid. So when we worked with this strategy in citrus, we used a model plant called Arabidopsis thaliana that allows for rapid screening of genes, and then we use this plant that usually does not produce or almost traces of caryophyllene. We added the gene for this compound, and then this plant started to produce 2,000 more this compound. So here are the three situations: the plant that produces just traces, that is the wild type, and then this is where the gene was removed from the plant, so no caryophyllene is produced, and this one that we produced to produce a lot of caryophyllene. We then tested it against the psyllid, and the plant that produced a lot of caryophyllene showed to be repellent to psyllid. So we identified the compound, the genes, and the pure compound was repellent, and then we produced that compound in a plant, and it was also repellent. So with those elements in hand, we had the basis to develop and go on with the project with citrus or oranges that were genetically modified to produce more of this compound with this repellent effect. So a whole strategy was set forth. And the steps of the process, well, after you have a plant generated at a lab, you have several steps. There's no time for going to the details, but we include like molecular characterization to see if the gene is present, number of copies of this transgenic plant, and then chemical analysis. They are essential for us to understand if the plant is producing a lot or a little of this compound, that is, if it's being modified, and then the behavior of the insect. That is key, but it's really time consuming in terms of detailed work to be carried out. So we have several days, like 10 days, and proper conditions for us to have results that is solid, because there's a lot of data that are not consistent in the literature. So this step of behavior of the insect is what will tell us if there is repellence or not. And we have to see if that's been changed or not, because we are changing the plant. We are having metabolic engineering of a plant, changing compounds that may be involved in production of hormones. So along these years, we generated 266 plants. That is a relatively high number of plants. Each of them, it's called an event of independent genetic insertion. So we had 7 strategies for combining genes. And all this metabolic change allows a lot of variation of genes and where we need them. I'm not going to go into details either. So that was done for Pera and Valencia oranges. So technology for genetic transformation with mature tissues, plants in the field already bearing and bearing fruit. Here's olfactometry with a smaller number of plants. If you were to perform olfactometry for all plants, it would take, what, 8 to 9 years for us to assess all the materials. Emission of volatile compounds is key to this project because that allows us to quantify how much of the repellent compound is in the air that is outside the plant. And that is exactly where the psyllid will notice that. Of course, we have visual characteristics that will attract psyllids to the plant, but also the olfactive characteristics that are key in our case here. Part of those plants is already in the field, and we are continuing with that for us to continue studying their behavior. Then we have events selected for plants that had good characteristics, and they're selected to go to the field very briefly for us to carry out repellence trials. Plants here are those that have the best characteristics. They have codes here, HB, GM1 until GM5. And they can release repellent compounds into the atmospheres up to 3,000 more times than a control plant. So the control plant will also emit that compound, release that compound, but in a really low amount. So we want to take orange that is naturally attractive to psyllid, and we want to turn that into a repellent characteristic to psyllid. So we want the plant to produce a lot of caryophyllene, that is the compound that we characterize as being repellent. Plants here with high production of this compound were tested as their repellents, and they all showed repellents to psyllid. Numbers, of course, are absolute. It's not 100% repellent. Repellents. We know that some in natural conditions in the field, maybe we cannot repel 100% of psyllid, but we can reduce at a certain amount, and we'll measure that in the field in trials with the psyllid, and at labs and greenhouse experiments we have we can do it, but it's we want to do that in the field, and then we can then potentially inoculate Liberibacter. So this is a field trial with all the biosafety procedures in this plot. So we have a cloud of repelling compound in this area to have decreased HLB in this area. So trials like this, we need structure and investments of a high level, and also we have to assess the results afterwards. And plants that have these repelling characteristics, where will psyllid find feeding material? Well, in a different plant, a plant that would not have this repellents, or maybe they can find, let's say, a myrtle plant in their way, like a border. A border of planted with myrtle would avoid psyllid. It doesn't like the repellents of citrus to go away or not even get into the grove. They will feed on myrtle and will die because of the application of insecticide. So the intention of using biotechnology here is to incorporate gene that is lethal lethal to psyllid, so they will die once, or Bergera once they, or even a citrus plant for validation. So psyllid will not reproduce or will die during their feeding on that plant. So ideally, our project will would believe in myrtle or bergera because they're not hosts for Liberibacter. There are plants that are not source of Liberibacter, but that then poses a different problem. That is the need to develop protocols to work with those plants in a lab. Protocols for genetic change, because there were plants that were not of interest to the scientific community. So we had to develop protocols for regeneration of myrtle in vitro, and then we also do that for Bergera. It's a little bit easier for Bergera than for myrtle, and then we have to go to the next step. That is the genetic transformation of myrtle, and this is the bottleneck of the project. Until today, we have been able to generate shootings with the characteristics of the genes that we want to insert into the plant, but we could not generate plants expressing that characteristic in a stable way entirely. So that has to do with changing genetically the plant, but we have to insert a characteristic that is lethal to the psyllid, so the growers do not have to constantly apply insecticide to control. So we want to use the technique of the RNA interference. Many of you may be familiar with that. That's used for controlling worms or borers, and that is effective to block the development of psyllid. So we have candidate genes that we can insert in myrtle to generate a compound that is double-strand RNA that is also produced by viruses and plants. So that is a technology that is employed by nature for proliferation. Only this way it would kill psyllid. Psyllid would not be able to reproduce in such plants. So that is a project that is very challenging, especially because of the absence of information that is basic for this crop. It's a rustic crop, this myrtle, Mariah. So for you to have this genetic transformation, we have to invest a lot of time. For the validation, here, if a strategy of the RNA is going to be then lethal to psyllid, we can use the genetic modification for citrus so that we do not wait until we can generate myrtle. So we are generating citrus with that gene, and then we are using the CTV vector. That is a strategy that for which Fundecitrus is the second lab in the world able to develop this strategy. That is, we use the citrus trichis virus that used to be a problem in citriculture, and today is all over the citrus region with the cross protection, especially for Pera. And we are able then to develop a CTV vector together with Professor Dawson in Florida. We were there for 4 years to cross this virus for this vector. That is a gene carrier to test a certain plant. The main advantage is that the citrus trichis virus, it resides in the phloem where psyllids feed, and the greening bacteria will also multiply. So this technology will also allow us to be to fight psyllid and the bacteria. So genes for killing the psyllid, for being lethal, is something that we have to test. And the third strategy that we are employing more recently is testing resistance against Liberibacter. So this panel was focused on psyllid, and we have a large number of pieces of information on that. And because of our inability to cultivate or to produce Liberibacter, then we have some limitation. But we do have some approaches to try and insert genes in citrus plants for us to hinder the multiplication of the bacteria in the plant. So we have here plants being developed, and then you're going to have in 2 years maybe the first test to present to the public. Strategies can be used jointly. So we have a strategy of attracting, repelling, or killing. We can have here a lethal border, for example, or if the border is effective, you can have a growth that is not even genetically modified with this border that is attractive and lethal. It's important to highlight that why we do not have this material, the management with the strategy that you are hearing about in the symposium should be implemented. What we need is what Professor Beauvais always mentioned. We have to have time to develop plans. So meanwhile, let's keep the management tactics for greening based on the 10 commandments that and the tripod for management of greening. Let's do that because in the future, if we have a resistant plant, then we're going to have a citriculture that is still vigorous in implementing this strategy. We have many challenges to come. So projects will involve a large number of collaborators within Fundecitrus and other institutions in Brazil, Spain, financial fund from FAPESP, or financial support from FAPESP. Here we have Leandro Penha as captain of this team. He's with us, a Zalki team, and a team that has already left us, like Professor Beauvais, that was paid a tribute yesterday. So thank you very much for your attention. We're going to be at your service for questions you may have.